Okay, if anyone can hear me, we will begin the class shortly.
I can hear you.
Okay. Now, I do have a few students. I just put out an announcement, so let's wait a while, see if students see the announcement. We'll begin shortly for the person who just added. Chapter 4. And we finish chapter 3. I think we did. We should be on chapter 4. Yeah, that's right. Okay, any questions?
Does this lecture still end at 6:20?
Yes.
Okay.
Okay. Let me share my screen. I'll be seeing my screen. We'll be covering chapter 4 today. Where were we? Yep. So we had talked about the gram-negative outer membrane. Any questions about any of that? All right, let's begin. For those of you who aren't logged in, you can see this online, and I'll put an announcement about that. But the gram-negative outer membrane does have a number of other molecules besides phospholipids and peptidoglycan. It also has lipopolysaccharides and lipoproteins. The gram-negative outer membrane does help protect the cell, and that's because this outer membrane of the cell wall can prevent things from getting towards the cell wall and then damaging the cell. And it can protect the cell from complement protein, which is something our body makes to help defend us. We won't really talk much about that, but it can also help protect the bacteria cell from antibiotics. And we did mention that penicillin G, for example, cannot get through this outer membrane. to then damage the peptidoglycan of the cell wall. The oligopolysaccharide can be as an antigen. So that oligopolysaccharide, part of the lipopolysaccharide, which is attached to the lipid A, can act as an antigen. And if you ever heard the name E. coli 0157H7, but 0157 is an antigen, which makes E. coli, or this strain of E. coli, a human pathogen. So E. coli 0157H7 will tend to make you sick, whereas normal E. coli does not. And then I did mention that lipid A is an endotoxin. That's something you should know. This lipid A is an endotoxin. So any patient that has a gram-negative infection, if that gram-negative bacteria has a cell wall, they'll have lipid A in the cell wall, which is an endotoxin. And we're not going to talk much about toxins now, but toxins are something which are toxic to us. So a gram-negative infection is generally worse than a gram-positive infection. Obviously, it depends if the gram-positive cell has the toxin or not, but if it doesn't, then the gram-negative cell will if it has a cell wall, because it at least has an endotoxin. And then there's the porin protein of the gram-negative cell, and that allows molecules to go through the outer membrane of the cell wall to either enter the cell, like for sugars and amino acids, or to leave the cell, like a molecule which is a waste product of the cell. If it can't leave the outer membrane, it will get trapped in the cell. And the waste molecule may be able to go through this porin protein that acts like a channel protein. All right, any questions about the gram-negative outer membrane? If not, let's compare the gram-positive cell wall to the gram-negative cell wall. Gram-positive cell wall has a thick peptidoglycan layer. The gram-negative cell wall has a thinner peptidoglycan layer, but if it has a cell wall, it does have a thin peptidoglycan layer. And that's more than one layer of peptidoglycan, which is the way your textbook shows it. It's actually many layers of peptidoglycan, but it's much thinner than the gram-positive cell wall. The gram-positive cell wall has a rigid structure to the cell wall, and the gram-negative, it's not as rigid. There is teichoic acid in the gram-positive cell wall, no teichoic acid in the gram-negative cell wall. There's no outer membrane in the gram-positive cell wall, and there is an outer membrane in the gram-negative cell wall, and it does contain lipid A in that gram-negative cell wall. All right, let's talk a little bit about the gram staining mechanism. Crystal violet iodine crystal complex forms in the cell wall. In the gram-positive cell wall, the crystal violet iodine does not leave the thick peptidoglycan in the decolorizing step. In the gram-negative cell wall, alcohol actually dissolves the outer membrane of the cell wall, and then the thinner peptidoglycan does not retain the crystal violet iodine, and so it washes out. And then the cell does stain pink when safranin comes in and stains the cell. This cell is showing you a summary of the gram-positive cell wall and the gram-negative cell wall. You only need to look at the top. Thick peptidoglycan in the gram-positive cell, thinner in the gram-negative cell, teichoic acid positive, present in the gram-positive, absent in the negative, outer membrane present in the negative, gram-negative, absent in the positive, and then lipopolysaccharide content is high in the gram-negative cell and virtually none in the gram-positive cell. There are some cell walls which are atypical. Let me first talk about the archaea. What we were talking about was the bacteria. The archaea have different cell walls. And so they are not like the cell walls we discussed previously. The archaea cell walls have cell walls made of pseudomurine. It's another disaccharide. It's not peptidoglycan. And some of the archaea have no cell walls around the bacteria. And in truth, there are a few. There's a family of bacteria that have no cell walls. We'll talk about that in a little bit. But the point is the archaea have different cell walls. It's curious, the archaea do have thick cell walls and bacteria or archaea that have thin cell walls. And the thick archaea cell walls stay gram-positive, and the thin archaea cell walls stain gram-negative. So they both stain gram-positive and gram-negative, but it's not for peptidoglycan in the archaea. It's from the thickness of the pseudomurine cell wall. Any questions about that? Among the bacteria, there are two families that have atypical cell walls. The mycoplasmas are bacteria that lack a cell wall. And they do have sterols in their plasma membrane to give the plasma membrane strength. And that's the same thing as animal cells have. They have cholesterol in their plasma membrane to give it strength. Well, the mycoplasma have other sterols in the plasma membrane to help give their plasma membrane strength. And how do you think the mycoplasma stain? Is gram-positive or is gram-negative? Nobody's taking a guess. They lack a cell wall, so they stain as a gram-negative. Anyways, they are a family of bacteria and they have atypical cell walls. The mycoplasmas, their shapes are pleomorphic, and it's because they don't have a cell wall to give the cell a shape. Another family of bacteria which has atypical cell walls, we've already mentioned this one, Mycobacterium. They stain acid fast because they have mycolic acid in the cell wall, and their cell wall is very different because they have this thick layer of mycolic acid. And they do typically stain gram-positive, and they are considered gram-positive because they do have a thick peptidoglycan layer, but they also have mycolic acid in the cell wall. And that's why they stain acid fast. The acid fast stain gets in the mycolic acid and doesn't wash out. Any questions about the mycobacterium? All right. You can damage the prokaryotic cell wall of a prokaryotic cell, making the cell susceptible to osmotic lysis. You don't need to know how you can do that, but you can do it with either an enzyme, lysozyme, which actually digests the peptidoglycan, or you could damage the cell wall with penicillin, an antibiotic, and it inhibits the bridge formation of peptidoglycan. If you have a wall-less bacteria, there's names for it. You don't need to know the names, but gram-positive cell wall, if it has no cell walls, they call it a protoplast. A gram-negative cell wall without the cell wall is called the spheroplast. You don't need to know those names. I'm not going to test you on that. But the gram-negative bacteria are not as susceptible to some agents which damage the cell, like penicillin G, because penicillin G cannot get through the outer membrane of the cell wall and then damage the peptidoglycan. There are some other antibiotics that are similar to penicillin G, like penicillin V is one that comes to mind, that also follow the same procedure, unable to damage the penicillin G. There are other antibiotics, however, which can damage gram-negative cells. And there are actually some antibiotics which only damage gram-negative cells. Streptomycin is the only antibiotic I'm going to talk about here. It is an antibiotic which is lipid soluble, and it can get right through the outer membrane of the cell wall. And so streptomycin is an antibiotic which can damage gram-negative cells. And there are other antibiotics. You don't need to know this other one, but oh gosh, losing the name of that one. Something B. It'll come to me. I'll try and think of it. There are antibiotics which can damage the gram-negative cell wall. Let's move on to talking about the plasma membrane. It's a phospholipid bilayer. It does have proteins in it. There's three types of proteins: peripheral proteins, integral proteins, and transmembrane proteins. Peripheral proteins are proteins that are either on the outside of the cell membrane or on the inside of the cell membrane. The other proteins, integral and transmembrane proteins, they span the cell membrane. The plasma membrane is mostly made up of phospholipids, and they form this type of arrangement where the phosphate heads are facing inside the cell because it's mostly water in the cytoplasm, and so the phosphate heads face the cytoplasm, and then the phosphate heads face outside the cell because there's mostly water outside the cell. If you think of it as a cell inside of blood, obviously the blood is mostly water, and that's true for actually the entire body is mostly water. So outside the cell is mostly water. And then the phospholipid fatty acid tails are on the inside of the cell membrane, and they take that on shape, blow this up, and so that the cell membrane is like this, with mostly phospholipids, although there are proteins in it. There are other molecules too in it, but we'll talk about proteins. The transport proteins will be a protein that spans the entire cell membrane, and then that allows a molecule to either enter the cell or leave the cell. When we talk about the plasma membrane, we can discuss it as the fluid mosaic model. This model states that the membrane is a fluid of phospholipids and a mosaic of proteins inside the phospholipids. It's hard to think about it, but my hand here is separated only by the cells, which are separated by a plasma membrane. And that plasma membrane is a fluid. It is a membrane that is as fluid as olive oil, so slightly a thick fluid. If you put olive oil in the refrigerator, it's solid. Our cell membrane isn't solid. It's as fluid as liquid olive oil. It does have proteins in it, and the proteins can move about to function. As you know, in AMP, if you take an AMP, receptor proteins, when they bind to a ligand, those proteins tend to come together, meaning they do move about in the cell membrane for the protein to function. And that's true of many proteins in the cell membrane, that they can move about in the cell membrane. The phospholipids themselves can rotate around as well as move around. And so they move around in the cell membrane. The only proteins that are anchored are proteins that are either anchored to the extracellular matrix outside the cell, like we can talk about a cell inside a bone. If that protein is anchored to the extracellular bone matrix, that protein won't move about. And the protein can also be anchored into the intracellular matrix. It's a matrix of proteins running around inside the cell. And if it is, then that protein is anchored and it doesn't move about in the cell membrane. But most of the proteins move about in the cell membrane. The plasma membrane has selective permeability. Shrink that, there we go. It has selective permeability, allowing the passage of some molecules, but not other molecules to pass. No large molecules can pass the cell membrane unless there is a transport protein specifically made to transport that molecule. Ions either do not pass the cell membrane, or if they do pass, they pass very, very slowly. And that is, ions do not like going through the bilayer of the fatty acid. A lipid-soluble molecule can pass right through the cell membrane. But if you think about it, most cells are surrounded with liquid, meaning a cell in the blood is surrounded with mostly water. And so there aren't a whole lot of lipids in the blood. But if a lipid-soluble molecule were to come up to the cell membrane, it can get right through the cell membrane. And then small molecules, especially smaller nonpolar molecules, can get right through the cell membrane, and they simply diffuse across the cell membrane. The most important small molecules are oxygen and carbon dioxide. And oxygen can diffuse right through the cell membrane, and carbon dioxide can diffuse out of the cell membrane. Any question about any of that? All right, the plasma membrane does have some specific proteins in it for photosynthetic bacteria. They have the pigments found in foldings of the cell membrane. So if it's a photosynthetic bacteria, the pigments for photosynthesis are in foldings of the cell membrane. And we call those either chromatophores or thylakoids, meaning it's similar to, thylakoid is similar to the chloroplast. There we go. But it is inside the cell membrane. That is also true that we can find the molecules that engage in ATP synthesis inside the cell membrane of a bacteria and the archaebacteria, but we're talking only about the bacteria, and that is the proteins that make ATP. In a bacteria, that doesn't happen in the mitochondria because they don't have a mitochondria. It happens in the cell membrane. If you damage the plasma membrane, you'll have the cell contents leak out, and that'll kill the cell. And you can damage the plasma membrane either with alcohols, quaternary ammonium compounds, and polymyxin antibiotics. This was the antibiotic I was trying to remember that damages gram-negative cells, polymyxin B. I knew I'd eventually get it, but that is an antibiotic. You don't need to know that, but it's one that damages the gram-negative cell. It does not damage the gram-positive cell wall. All right, any questions about the plasma membrane? If not, let's talk a little bit about movement of materials across membranes. This applies to the plasma membrane. And then for eukaryotes, it applies to any organelle that has a membrane around the organelle. There are two processes that allows molecules to move across a membrane. There's a passive process where the molecule moves down the concentration gradient. So the molecule moves from areas of high concentration to areas of low concentration. The passive movement of material can happen in three different ways. There's simple diffusion, where a solute moves down its concentration gradient. For example, we can talk about oxygen moves from areas of high concentration outside the cell to areas that it's lower inside the cell by simple diffusion. There's also movement of material down the concentration gradient by facilitated diffusion. That's where a solid molecule moves across the cell membrane, for example, by using a transport protein. One that we'll talk about in just a little minute is fructose moves across the cell membrane by a transport protein, and this happens by facilitated diffusion. And then the third way that molecules can move passively across a membrane is by osmosis. Osmosis is where a solvent, meaning water in living systems, moves across the cell membrane, moving from areas of high concentration to areas of low concentration. So osmosis is specifically the movement of water across a membrane. And water is a solvent, not a solute. So if it was a solid, it wouldn't happen by osmosis. It would happen by either simple diffusion or facilitated diffusion. Any question about any of that? The other way that movement may occur across the plasma membrane is an active process where the molecule moves against its concentration gradient, meaning the molecule moves from where it's lower in concentration to where it is moving higher in concentration. This happens with a transport protein, specifically pumping the molecule generally into the cell, but it could pump it out of the cell as well. And that transport protein has to use a molecule of ATP to transport that molecule against its concentration gradient. For example, if we have not ATP, if we have an amino acid outside the cell is going to want to transport that amino acid inside the cell, even though there's higher concentration of amino acids inside the cell. So that cell will use a transport protein to pump that amino acid into the cell. And for each molecule that's moved into the cell, the transport protein will have to use up a molecule of ATP. Any question about any of that? All right, let's talk a little bit more about simple diffusion. There's the movement of molecules against its concentration gradient. Movement of molecules down its concentration gradient, where a solid moves from an area of high concentration to areas of lower concentration, such as we put a drop of dye in the water. Simple diffusion is the way that oxygen and carbon dioxide move into a cell or out of a cell. We also have facilitated diffusion, where a transport protein moves the molecule either into the cell or outside the cell. Facilitated diffusion transport proteins happen where the molecule moves from areas of high concentration to areas of low concentration. So facilitated diffusion does have a transport protein, but this transport protein does not need to use ATP to move the molecule across the membrane, because the molecule is moving from areas of high concentration to areas of low concentration. Okay, so the molecule is moving down a concentration gradient, and you can think of this as a channel protein, because this molecule is not really pumping it across against this concentration gradient. Any question about any of that? An example of a molecule moving by facilitated diffusion using a transport protein is the molecule of fructose. So when you're eating high fruit diet, having a lot of fructose, the molecule of fructose will move into the gut cells by facilitated diffusion. But once the fructose concentration inside the cell equals the concentration outside the cell, then the molecule of fructose can no longer move into the cell. So that means that all the fructose will not be digested. Any question about any of that? And that is, it will move into the cell until the fructose inside the cell equals the fructose outside the cell. That's different from glucose, and that is every molecule of glucose outside the cell will want to transport it into the cell using a molecule of ATP. And so if you eat glucose, all of that glucose will be moved from the gut into the cells of the gut. All right. Let's talk a little bit more about osmosis. When we're talking about solutes being transported across the membrane, that happens either by simple diffusion or facilitated diffusion. But when we're talking about water, water moves by osmosis. Water is a solvent, not a solute. And osmosis happens from water moving from areas of high concentration to areas of low concentration. Now, when we're talking about water moving across the cell membrane, you should realize that we use the solute to define what is high and what is low. And the solute, if it is low in concentration, meaning lower outside the cell than inside the cell, then the water is the reverse of that. The water will be high outside the cell where the solute is low. And where the solute is high inside the cell, the water will be low. So water will be opposite of the solute. And I'm telling you that we define this by the concentration of the solute. And we discuss that as isotonic, hypotonic, and hypertonic. And these are defined by the concentration of the solute. Where the solute concentration is equal, that is isotonic. Concentration outside the cell is equal inside the cell. And so there's no net movement of water into and out of the cell. When I'm saying no net movement, if water does move into the cell, one water molecule, then one water molecule moves out, but the net movement of water is equal. Okay, and the cell is happy in an isotonic solution because there's no net movement of water, and that means no water will leave the cell, no water will enter the cell, and so the cell is happy. There's also hypotonic solution and hypertonic solution, where each is defined by the solute concentration outside the cell relative to the solution inside the cell. In a hypotonic solution, there is more water outside the cell than inside the cell, and so water will tend to move inside the cell, causing the cell membrane to expand. If there is no cell wall around the cell, then that cell membrane will expand until the cell bursts, and that would damage the cell and destroy the cell, and the cell would burst by osmotic lysis. However, the cell wall can protect the cell membrane, and water will move into the cell up until the cell membrane comes up against the cell wall. And then the cell membrane can't expand any further, and that will limit the water moving into the cell, meaning no more water can move into the cell. Most bacteria live in a hypotonic solution. And if the cell wall is damaged, like by penicillin G, the bacteria will lyse because the penicillin G damages the cell wall. All right, any questions about hypotonic solution? If not, let me talk about the last one, the hypertonic solution. That is where the concentration of water is less outside the cell than, let's see if I got that right. Yes, less outside the cell than inside the cell. So there's the net movement of water outside the cell. And remember, where the water is low, the solute concentration is high. And so water will move outside the cell. And that may cause the cell to die by plasmolysis, but the water will leave the cell, and so the cell membrane will shrink. This generally leads to cell death, and that is why when you have a bottle of molasses and bacteria fall into it, the bacteria can't grow. It's because the water leaves the bacteria and then goes into the molasses. And that's why molasses doesn't spoil. Most of the cells that fall into molasses will die by plasmolysis. Any question about any of that? If not, I'm going to stop here and I'll see you at 6:30 in the lab. All right, I'll see you at 6:30.